In a corner of Austria, which now falls in the present-day Czech Republic, in a monastery there was a monk called Gregor. He was none other than Gregor Mendel, who was the pioneer of genetic studies. So what exactly did he do? Being a student of both science and math, he studied pea plants. In the garden of the monastery, he planted several pea plants and mixed and matched different characteristics to study them. The pea plants he studied had many variations, like different seed colors and length of the plant, for example. Constant observation for many years led him to conclude several things about the pea plants. With this, he wrote his paper in the year 1865 titled Experiments in Plant Hybridization. Mendel realized that there was a certain pattern of plant reproduction in all his experiments. These observations went on to be his famous laws. What exactly were these laws? To study the laws, we will first have to understand a few basic concepts. We have studied that a cell contains a nucleus. What did we study about the nucleus? That's right, the nucleus of a cell has a set of chromosomes which contain the DNA. DNA, as we know, is the deoxyribonucleic acid which contains the genetic information. What is DNA actually made up of? DNA is made up of bases which encode specific information for the making of proteins. The two bases that usually join each other are called a base pair. These bases will be joined to several other bases in a specific pattern. When these bases go against the standard pattern, we call this a mutation. The two bases that usually join each other are called a base pair. Mutations can sometimes affect one base pair and other times an entire segment. What would happen if it affects an entire segment? This means that there will be several base pairs which will undergo change. So, let us look at what we have studied so far. The bases are the basic fundamental units of a cell. They join some other base and form a base pair. Several such base pairs will form a double helical structure. They are two strands twisted around like a ladder. Do you know what we just formed? Well, we call this the DNA molecule. What will several such DNA molecules together form? DNA strands, when tightly wound and coiled, will form a chromosome. So if we look closely at a chromosome, it is nothing but a long strand of DNA tightly wound and coiled to form this shape. But the chromosome seems to have two individual strands joined at the center. What is each single strand called? One single strand is called the chromatid. Two chromatids join at a point in the center to form a single chromosome. This center point here has a nice name. Do you know what it is called? It's called the centromere. Well, do all chromosomes look like this at the very beginning? Are these two chromatids one and the same? Do they have the same DNA and the base pairs? Watch our next video for the answers.